Hello everyone and welcome back to BioBytes, where big biology becomes bite-sized. Today we are diving into one of the most fascinating and hardest working structures inside every cell. So let me start with a small guesswork. I don't have a brain, yet I am the sharpest worker in the cell. I am made of RNA and proteins. I come in different sizes. You will find me everywhere in all living organisms like bacteria, plants, animals, and even fungi. And my main function is I am a protein builder. And a single cell contains thousands of me, working 24 by 7 without even taking a break. I build proteins. Can you guess who this is? Yes, you are correct. So let's explore the one and only ribosomes. So it is our ribosome which will work continuously and make proteins. Absolutely correct. So let us learn about ribosomes in our class today. Right. Now, as you all know, children, ribosomes are the protein factories. Their main function is they produce or they synthesize or they manufacture proteins. Now, for the first time, ribosomes were discovered by a scientist called as George Palade. Right? So, a scientist by the name Palade, he discovered ribosomes formally, but it was first observed by Claude in 1941. But Palade was the one who identified it and named it as Palade particles in 1955. Right? And these particles were so small that they looked like small dots. Right? And then in 1958, a scientist by the name Richard Roberts. So a scientist by the name Richard Roberts gave the name ribosomes. Right? Earlier they were called as microsomes, but later on Richard Roberts called them ribosomes because they were rich in RNA, ribonucleic acid. Right? So he named as ribosomes. Right. Now shall I tell one interesting fact? John Palade won the Nobel Prize in Physiology or Medicine in 1974 for his discovery. So Palade, who gave the name Palade particles, he won the Nobel Prize. So the little dot he found changed life forever. Oh, okay. So let us learn about these sites of protein synthesis, the engines of the cell. Now, these ribosomes are universally present. They are present in all prokaryotes and eukaryotes. They are found freely in cytoplasm, and we have learned they are also seen on the RER, that is a rough endoplasmic reticulum, right? So they are attached to it and they are present in the mitochondria and the chloroplast also. Now, if it comes to types of ribosomes, children, remember ribosomes here basically they are of four types. Here you can see they are 70S and 80S type of ribosomes. So here you can see 70S and 80S. Now 70S are mostly found in prokaryotes or bacterial cells, right? And 80S are found in eukaryotes, in all plant and animal cells, and they are they are free ribosomes, they are seen on the RER, right? And 60S ribosomes are mostly found in the mitochondria, and 77S ribosomes are found in fungi. Right. Now, these ribosomes, you see, the ribosomes, when you see the structure, they are made up of two subunits. There is a larger subunit, there is a smaller subunit. Right? So, together, so the ribosomes are made up of two subunits. And these subunits, we measure them by Swedberg units, SVEDBERG. Now, we just identify them by their Swedberg unit. That is the way they get precipitated, or the way they get concentrated at the bottom when we put in a centrifuge machine. So their sedimentation coefficient is measured by Swedberg units, and that is what we call them as 70s, 80s when I am telling. So this S is nothing but it is Swedberg units. So this is a unit, Swedberg unit. And what is actually this? So you will see this 70s or 80s type of ribosomes getting sedimented in the in at this particular rate when we put in a centrifuge machine. Right. Now, shall I tell you one interesting fact? A single mammalian cell, if you see in a single mammalian cell, in the cell or animal cell, you will find nearly 10 million ribosomes in a single mammalian cell. So you see how fast the protein synthesis is taking place and how adequate it is in our animal cell. Right. So you can see ribosomes are like workers sitting on the factory shelf. They make proteins, they pack it and ship it out of the cell. And free ribosomes are like freelancers. Okay. Right. So now children, let us learn now let us just dive into the detail of this 70s and 80s type of ribosomes. Like what are they? Again, what is this big piece and what is this small piece, or the larger subunit or the smaller subunit, what you see. Right. So now you see children here the subunit. So when it comes to the ribosome, they have the larger subunit, the smaller subunit. The subunits are made up of rRNA and proteins. Right. And remember, the large subunit and the smaller subunits, they come together at the time of protein synthesis. Right, when the proteins are being manufactured or they are prepared, at that time you find these two subunits joined. Right, they sandwich, and between them you have the proteins being manufactured. So how these subunits work is, children, you can see both the subunits are together, and you can see together we call it as a dimer. Right, the two subunits associate to form a dimer, and these dimers attach to a single mRNA. So this is a mRNA, right, and to form a polyribosome. So this you call it as a polyribosome. Many ribosomes, they are attached, they form the dimers, and they are making a what I should say, the proteins. Clear? So if you see the mRNA position, so the larger subunit, the smaller subunit, between the two subunits you have the mRNA which runs. Right? And you can see here the position, it is perpendicular. Right? So ribosomes are perpendicular to the mRNA. It's like this. And let me tell you, magnesium plays a major role. Magnesium, the concentration of magnesium will strengthen the strengthen the dimer to come together and the formation of proteins. Right. So now here if you see the 70s or the ribosomes which we find in prokaryotes, the 70s ribosome, right, it has a bigger subunit of 50s and a smaller subunit of 30s. As I told you, the ribosomes are made up of rRNA and proteins. Right? Now, the rRNA composition in this is, if you see the 50S, right? It has the rRNA composition of 23S and 5S. Clear? So this is a rRNA composition seen in the larger subunit. Now the rRNA composition seen in the smaller subunit is 16S. Right? Children, you need to remember one more thing I want to tell you all. If you see the 70S ribosome, the 70S ribosome is made up of 50S plus 50S and 30S. Now most of the students have a doubt here. Now 50S plus 30S is 80. But how come this is 70S ribosome? Remember children, the values of the larger and smaller subunit is not additive. It is not additive. No, it is not additive. It depends on the sedimentation coefficient. right? It's not based on addition. So this is very important. In most of the competitive exams they will ask what is the what are the larger and the smaller subunits of 70s ribosomes. So 70s ribosomes, it is made up of larger subunit of 50s and smaller subunit of 30s. Right, it is 70s. And what is the rRNA composition of 50s? It is 23s plus 5s, right? And smaller subunit, it is 16. Now when it comes to 80s ribosomes, you see the 80s ribosomes, the 80s ribosomes. The 80s ribosomes are made up of two subunits. The larger subunit in 80s ribosomes, you have the larger subunit which has 60 Swedberg unit value, and the smaller subunit has a unit value of 40 Swedberg units, right? So I am repeating, it is not additive. 60 plus 40 is 100, but it is not additive. It is based on the sedimentation coefficient. So what are the subunits of 80s ribosome? The subunits are 60s plus 40s. Now in this 60s and 40s, if you see the rRNA, let me repeat, our subunits are made up of rRNA and proteins. The rRNA composition in 80s is 28s plus 5s, and there is one more you can include, it is 5.8, right? 20s, 5s and 5.8s, right? So these are this is the rRNA composition seen in 80s ribosome. And in smaller subunit, the ribosome is 18s, 18 Swedberg unit, right? Now just remember that the 60s and 77s, they are present in the mitochondria and they are present in the cells of fungi, right? So in mitochondria, the ribosome which is present, it has only the larger subunit which has the rRNA composition of 28 plus 5s, right? And the 77s which is present in the cells of fungi, it has larger subunit of 50 Swedberg unit and smaller subunit of 27 Swedberg unit, right? Okay, so now hope this is clear. So in this, what you need to remember about the larger and the smaller subunit of the types of ribosomes. So in 70s you have 50s and 30s, and in 80s, which is seen in eukaryotes, you have 60s plus 40s, right? Hope this is clear, right? Now when it comes to chemical composition, as I told you, ribosomes mainly contain RNA and proteins, right? So the RNA is nothing but the rRNA, ribosomal RNA, right? And the proteins. Now when you see the ribosomal RNA in 70s, you have 65% of ribosomal RNA, rest are the proteins. In 80s ribosomes, you have 45% of ribosomal RNA and rest are proteins, right? So you can see here the proteins are again, so this is the rRNA and these are the proteins. The proteins are again of two types, core proteins and peripheral proteins. Core proteins are tightly bound, peripheral proteins, they are loosely bound. Clear? And these are basic or acidic in nature and mostly they act as enzymes. Here some scientists also regard this as ribozymes, because ribosomes also act as enzymes. So we also call them as ribozymes because they contain the ribosomal proteins which act as enzymes too. Right. Now when you see the role of ribosomal proteins, mostly what the ribosomal proteins do is they hold the RNA in a specific three-dimensional structure. They hold the RNA. You can see the RNA is held in a structure, three-dimensional structure, and they provide stability to the ribosome. They act as enzymes, peptidyl transferase in the larger subunit. I told you they act as enzymes. They interact with proteins called elongation factors that help in proteins. As the proteins are prepared, as the proteins are being manufactured. These ribosomes, the ribosomal proteins will interact with proteins called as elongation factors, EF, right? And these elongation factors are responsible, or they help in protein synthesis, right? So ribosomes are the site of protein synthesis. And what are the ribosomes composed of? RNA, rRNA and proteins, right? So you can see here how on the Golgi body, sorry, not on the Golgi body, it is on the endoplasmic reticulum we have seen RER. On the rough endoplasmic reticulum we have the ribosomes, which help during which synthesize proteins, right? Okay, the next. Okay, now just a small this one. As I told you, the rRNA, the ribosomal proteins, they act as enzymes. And the ribosome is essentially a giant RNA machine. And the person who discovered this and brought it to the world, right? It was Venkatraman Ramakrishnan. Yes, it was Venkatraman Ramakrishnan, this scientist. And there was another scientist by the name Thomas Tids. Another scientist by the name Thomas. And another scientist by the name Yonath. So these three scientists, they spoke about the ribosomal proteins, right, which are like giant RNA machines. And these three scientists won Nobel Prize in the year 2009 for their discovery, right? So the rRNA is like an engine. The ribosomal RNA is like the engine, and the proteins are like the frame of a car. So the frame will support, but the engine actually does the work. It's like that, right? So now now when it comes to, let us talk about the non-ribosomal proteins. So there are some non-ribosomal proteins, or what we call them as factors, ribosomal factors, right? Now these non-ribosomal proteins, they mostly assist in initiation, elongation and termination of protein synthesis. Okay, so now let me explain you this. Each ribosome, children, it has three functional sites, right? So you have the A site, P site and E site, right? So let me tell you the A site, P site and E site. A site is nothing but it is the amino acyl site. A site is amino acyl site, and P site is peptidyl site, right, where the growing polypeptide chain is held, right? You can see here, right, the growing polypeptide chain is held. This is the first A site. The growing polypeptide chain is held. This site is called as the P site. P site also called as peptidyl site. And the last site where the empty tRNA exits, so here the exiting one, right, where it exits, we call it as E site, right, where we call it as exit site. So you can see, based on this, if you remember this, you will remember the types of factors. Initiation factors, which help in binding of mRNA to the ribosomes. You can see here initiation factor, it helps in binding, right? IF, initiation factor, helps in binding. Elongation factor is the amino acyl, or the you see here, right, the way the elongation or the protein synthesis takes place. right? They assist in translocation. Translocation means the protein synthesis process. And then we have the termination factors. Termination factors means the exit site, right? So the termination factor is where the empty tRNA exits after giving up its amino acid, right? So just remember APE. APE, A is entering, P works, and E exits, right? So you need to remember this. The next we have, these are the three protein factors: initiation factor, elongation factor, termination. Initiation in the sense entry, here it is processing or working, here it is terminating or release factors, right? Now when it comes to this origin of ribosomes, now children, these ribosomes are synthesized, these ribosomes are produced in the nucleolus, right? So the nucleolus contains ribosomal DNA, right? Ribosomal DNA, and it is transcribed by RNA polymerase enzyme to form a precursor called rRNA, right? So what happens actually is the nucleolus contains rDNA, ribosomal DNA, right? Now this ribosomal DNA, it is transcribed by RNA polymerase, right? And from where it is transcribed? From rRNA, that is a 45S in eukaryotes, right? Now this rRNA is processed and then it is cleaved into smaller RNA. You can see here the 45S pRNA, it is cleaved here, and it is cleaved to form the 28S RNA. In eukaryotes, you see you have 60 and 40, the 60 subunit. In 80S RNA, if you see, you have 60 subunit, you have 40 subunit. In that 60 subunit, you have 28S, 5.8, and 18S. The smaller subunit has 18S rRNA, and the larger subunit has 28S and 5.8S and 5S also, right? Okay, so now this rRNAs associate with ribosomal proteins in the nucleolus to form pre-ribosomal. So these rRNAs, what are formed here, these will get attached with the proteins which are made in the cytoplasm, and these are brought to the nucleolus to form pre-ribosomal subunits, the subunits. These subunits are again transported to the cytoplasm, and in the cytoplasm the bigger and the smaller subunits form the functional ribosome. So it always starts in the nucleolus and continues into the cytoplasm, and by the time it reaches the cytoplasm, the two subunits associate on mRNA to form functional ribosomes ready for protein synthesis. Right. Now, the functions of ribosomes. The major functions of ribosomes, they act as, actually now we have learned protein synthesis, right? They catalyze a peptide bond formation between amino acids, right? Peptide bond, how the amino acids are made into proteins, you see, through the peptide bond. Then they associate with mRNA and tRNA to form the functional translation complete. So they associate the messenger RNA, the tRNA, they associate, and then the translation is completed. And all the proteins are synthesized by the ribosomes. And these proteins required for cell growth, repair, all these are synthesized by the ribosomes, right? And the free ribosomes synthesize proteins for use in the the proteins which are used in the cytoplasm, which are bound by the rRNA, right? All these, you see, they are they are the proteins for secretion, membranes, and lysosomes, right? Now these ribosomes, they are very essential for our life, because protein synthesis plays a major role in cell growth, repair, and other functions. For all cellular activities, we need proteins, right? So all the cellular activities will come to a standstill if these proteins get affected, right? So now let us come back to what we have understood. The question, who gave the name ribosome to the cytoplasmic particles? So it was discovered by Palade, but it was the name was given by Richard Roberts, right? So remember, R for ribosomes, the name given by Richard Roberts. Which of the following correctly represents the subunit composition of a 70S ribosome? 70S ribosomes, we have learned it consists of two subunits called 50S and 30S. Right? So it is C. I told you it is not additive, children. It is not additive. It depends on the sedimentation coefficient. Right. Then next. Which antibiotic targets the 30S ribosomal subunit and causes misreading of genetic code? Right. Now here the antibiotic, let me tell you about this. There is an antibiotic in this. Now suppose I have a statement here. The cat ate the rat. Okay. Now suppose if I misread this. The cat ate, instead of rat, instead of this R, I use bat. The cat ate the bat. So the entire sentence changes. So you see, just one word, one letter I should say. So similarly, there is an antibiotic which acts on the smaller subunit 30S, right? And that antibiotic is streptomycin. Remember, children. So this streptomycin, this antibiotic, they all work for bacterial infections. You know this. How this antibiotic works is, in bacteria, this antibiotic, it will misread this instruction, this genetic code, right? And it will misread when it is done, when something is done wrong here, misread. So there is a mistake done here. So when there is a mistake, naturally the cells will not divide, they will not multiply, and the cell will die. Right? So this is one of the strengths of antibiotics. So that antibiotic streptomycin, it targets the smaller subunit, the 30S ribosomal subunit. Whereas this erythromycin, just remember children, erythromycin and chloramphenicol, they target the larger subunit. Right? And this also targets the larger subunit, the 50S. Right? 50S they target, but this streptomycin targets the 30S ribosomal subunit, causes misreading and death of bacteria. Okay. Which of the following ribosomal RNA is found in the small subunit of ATS? In ATS, what do we have, children? In ATS, we have 60S plus 40S. So in the smaller subunit, which is a rRNA we see. The rRNA we see in ATS is 18S rRNA. Right? So 18S rRNA is seen in the smaller subunit. Right. Now this is one more. Diphtheria toxin inhibits protein synthesis. Protein synthesis means production of proteins. Right? Now here there is a small clue through which you can go to reach the answer. Now cleaving is what, children? Cleaving means to break. Right? But here in the question, what are they asking? In the question they are asking protein synthesis. That means production of proteins. Right? So we are not talking about breaking. Wrong. Inhibit, EIF2. E eukaryotic. I stand for initiation. We are talking about protein synthesis. Right? That means we are talking about elongation factor. We are not talking about the initiation or termination. Right? So inhibiting, no. Right? Then blocking the E site. E site is a last exit site. This also is not. Protein synthesis means where the proteins are being manufactured, this phase. So this gives us a clue. Right? Initiation factor 2. So ADP ribosylating, initiation factor 2. right? So you have initiation factor 1, initiation factor 2, initiation factor 3. Just remember this diphtheria toxin, it inhibits the protein synthesis in eukaryotes by this particular protein ADP ribosylating EF2, right? Okay. In eukaryotes, the 45S pre-rRNA transcript is processed to yield which combination of mature ribosomal RNAs? So, in 45S pre-rRNA, remember children, you have 28S, 18S and 5.8S. Right? So you have 28S, 18S and 5.S. So you just need to remember the composition of this ribosomal RNAs. You need to remember. Right? So just remember that it is 28, 18, 5.8. Clear? So this is the answer. This is all about today's class on ribosomes. Keep practicing, keep learning, and be happy. Thank you.